Today we are going to discuss about the structure of chromatin. Basically, we know that chromatin is a nuclear protein which is made up of the deoxyribonucleoprotein, including the deoxyribonucleic acid, DNA, as well as the basic protein known as histone. Now, histone is a protein which is rich in arginine and laminin. Arginine is a positive and laminin is a negative amino acid. Both are richly present inside the histone, and these histone basically are also known as a packaging protein. These package the chromosome. These package the chromosome material. These package the DNA in such a manner that DNA is having a coiled structure. It is having a helical structure. So in order to not become coiled or form a network, bunch of networks within the cell's nucleus, the uh, histone present inside the chromosome prevents the DNA to become saturated, become form a bunch of networks within the nucleus, due to which histone is present as a packaging protein to avoid collapsing the DNA within DNA in order to form several unseparable networks. So in that case, this is how this is why the histone is present. Secondly, histone is basically involved in maintaining the acidity of DNA. As DNA is acidic, it is a nucleic acid, so stone is basic. So when both of these combine, they form a neutral structure, a neutralized structure in which uh, the nucleus ultimately becomes neutral, neither acidic nor basic. Now when it comes to the structure of chromosome, it is in such a manner that here we are having uh, this is the basic structure of chromosome. Basically, the chromosome is covered with an outer layer which is known as pellicle. Chromosome is covered with an outer layer which is known as pellicle. Apart from which, inside the inner layer, there is the presence of matrix. There is a presence of fluid or matrix. Inside the matrix, there is a double-stranded DNA. There is a double-stranded DNA, and that double-stranded DNA is present in one chromatid. In one chromatid, there is one double-stranded, complete double-stranded DNA. Apart from which, in a complete chromosome, there are two double-stranded DNA. Secondly, the chromosome consists of three parts: the centromer, chromosomal arms, and chromatids. The part which starts from the center of the chromosome, the part which starts from the center of the chromosome, oops, it's there. So the part which starts from the center of the chromosome to the terminal end is known as the chromosomal arm, and the chromosomal arms are connected with each other at a central part which is known as centromer, and that centromer is also known as primary constriction. It is constriction, depression present inside the chromosome, which is known as primary constriction, which connects several chromosomal arms. On the other hand side, this complete chromosome, for suppose this is a chromosome, within that chromosome, this complete arm, this complete two set of chromosomal arm connected with a centromer is known as the chromatid. Now these two chromatids which are present inside a chromosome are the sister chromatids, and on the other side, if there are two different chromosomes, then this arm and this arm will be collectively considered as the non-sister chromatids. Now, apart from this, there is a primary constriction here. As you can see, primary constriction or centromer, where the chromosomal arms are joined, and over the primary constriction, there is kinetochore disc-like protein. Apart from which, there is a secondary constriction in some chromosomes, such as in humans on the 12th, in humans 12th, 13th, 14th, 21st and 23rd chromosome is having secondary constriction, and that secondary constriction is present near to the end, and that contains a region which is known as NOR region, a nucleolus organizing region, from where the nucleolus is synthesized. Apart from which, the this region which is formed by the secondary constriction is known as the set chromosome, and that set chromosome consists of satellite DNA. That satellite DNA Basically, uh, this satellite DNA is a junk DNA. Why is it a junk DNA? Because it consists of heterochromatin region in such a manner that this heterochromatin region is having transcriptionally inactive genes which have no use in the cell in transcription. Secondly, they are having repetitive sequence, due to which they are having transcriptionally inactive genes and repetitive sequence, due to which that causes to make them junk or useless within the cell. So these are useless DNA, junk DNA, due to repetitive sequence and heterochromatin region having transcription inactive gene. Apart from which, when it comes to chromosome, these the upper arms are known as the P arms and downward arms are known as Q arms. And on the basis of the position of centromer, there are different types of chromosome. For suppose a chromosome which is having the centromer position in equally in the center is known as metacentric chromosome. Apart which is having the centromer position in the center and all arms are equal. Secondly, if there is a chromosome which is having the centromer position near to the center and the arms are unequal, then it is known as submetacentric chromosome. In humans, mostly submetacentric and metacentric chromosomes are found. Secondly, if we are having a chromosome which is having one arm short and other arm exceptionally long, then this type of chromosome is known as subtelocentric or acrocentric chromosome, which is having its centromer near to the end. And fourthly, there is telocentric chromosome, which is not having the P arms, it is only having the Q arms and the centromer is present at the end. Telocentric chromosome is not found in humans, and there are chromosomes which are acentric, which are not having any centromer, which are found in case of abnormality. Bicentric, which are having two chromosomes, which are having two centromers. Polycentric, which are having many centromers. In that case, this is classification. Apart from which, when it comes to chromosome number is different in different species. For suppose in corn, that is 20, glycine maize, that is 20, and sweet pea, pisum sativum, it is 14, and a dragonfly, sorry, in Pisum sativum, it is 14, is also known as sweet pea or garden pea, and fruit fly, which is also known as Drosophila, which is also known as Drosophila melanogaster. Drosophila melanogaster is its scientific name. Drosophila melanogaster. Fruit fly is having its chromosome number 6. Apart from which there is wheat having 42, cotton 52, as well as there is sugarcane 80, humans 46, and in frogs 26. Apart from which, these are the number of chromosomes and present in several organisms. And that's all about the video. Hopefully, we'll meet again in another one. Till then, have a good time.